Hello and welcome everyone to ATGC of Genetics. Today we'll be talking about ATGC of Spinal Muscular Atrophy. Let's just quickly go through what all we'll be discussing in this video. We'll be starting off with an introduction to spinal muscular atrophy. We'll be talking about what is the cause of SMA, the spinal muscular atrophy. We'll be discussing its different types and their symptoms, inheritance of SMA, diagnosis, and its treatment. So let's get started. What is spinal muscular atrophy? It is a motor neuron disease, which means that it affects the ability to move. It is characterized by muscle weakness and a progressive degeneration, which means that it can cause problems in movement, such as sitting up, walking, swallowing, and sometimes even breathing. It is genetic in nature, which means that it is something which you are born with. Although its age of onset could vary, but if we are born with the gene, it is pretty much sure that you will be developing it sometime later in life. Based on the age of onset and its severity, it's divided into different types. SMA is also known as 5q SMA muscle degeneration, and spinal amyotrophy. So as I said before, that it is a motor neuron disease. So what does it mean? From the word motor, you can make out that it is something to do with movement, and it affects the ability to move. As the name also suggests neuron, it means that there is some problem in the neuron, which are the cells which are responsible for conveying the signal from brain to the muscle and help in movement. It's also important to note that in a person, the cognitive function, the thinking process, emotions, everything is perfectly fine. The only problem is with the movement. Let's now understand how do we move our volunteer muscles, which are the muscles we move on our own will. As you can see in the diagram right here, that the brain sends signals to the spinal cord, which then conveys the signal to the muscle via the motor neuron, and thus our muscle moves. So what happens in SMA? In an individual who is affected with SMA, the brain again conveys the signal to the spinal cord, but as we discussed earlier, that it is a motor neuron disease. So there is a problem with the motor neuron, that is, they are damaged. Thus, the motor neurons are not able to convey the signal to the muscles, and thus the process highlighted here is affected, it is not carried out, our muscles do not receive the signal and thus are unable to not move, and this results in their progressive degeneration. It is also important to note that the lower limbs and the proximal limbs, which are the limbs which are closer to the center of the body, such as hips, shoulders, are more affected. Let's just do a quick recap and a breakdown of the name of the disease. Why is it called SMA, or spinal muscular atrophy? It's called spinal because there is an irreversible loss of the anterior horn cells in the spinal cord. Thus, the motor neurons of the lower portion of the anterior spinal cord are affected. It's called muscular because its primary effect is on the muscles. And it's called atrophy, which is the medical term for shortening or weakness, as seen in the muscles in spinal muscular atrophy. It's also important to note that 1 in 50 people worldwide are carriers of spinal muscular atrophy, and approximately 1 in 6,000 to 10,000 cases per live births are observed. August is Spinal Muscular Atrophy Awareness Month. Let's now quickly go through the pathogenesis of spinal muscular atrophy and see how it is caused in our body. As we discussed earlier, that SMA or spinal muscular atrophy is genetic in nature. So let's now quickly look at the genetics of this disease. You might know that DNA or deoxyribonucleic acid is the genetic material of a body. You can imagine DNA as a pearl necklace. You can see that there are many different pearls embedded in the necklace, and you can imagine each pearl as one gene. So you can see that there are a lot of different genes on a DNA, and the gene could be anywhere. It could be hair or it could be hair. If you do not know what a gene is something which produces a particular protein in our body. This could be any protein which is utilized in various different ways in our body. For example, a gene might be responsible for producing insulin, which is utilized for breakdown of sugar. So let's just assume the second circle is the SMN1 gene, which is the survival motor neuron 1 gene. From the name itself, that is survival motor neuron, you can make out that this protein which is produced by this gene is vital for the survival of motor neurons. So let's zoom right into it and understand it a bit more. As you can see in this diagram, that the SMN1 DNA or the gene finally gives rise to the SMN protein or the survival motor neuron protein. In this diagram again, you can see that a gene gives rise to a protein. And it's important to note that for any gene, and even for the SMN1 gene, the gene is made up of introns and exons. You can imagine this as a train which is made of different compartments or coaches. So the different exons and introns in a gene. As you can see from the diagram as well, that only the exons join together at the end to form the protein. So what happens in the SMN1 gene which is mutated or which is disease causing or which causes spinal muscular atrophy is that the 7th exon is missing, which then gives rise to a non-functional protein. As this protein is not functional, and as we discussed earlier, it is vital for the survival of motor neurons. The motor neurons begin to shrink and die. It's also important to note that right beside the SMN1 gene is the SMN2 gene, which is also important in discussion of spinal muscular atrophy. So let's talk about it as well. This gene is 99% similar to SMN1 gene. It also produces SMN protein, but here only 10% of the protein produced is functional. A person can have anywhere between 1 to 8 SMN2 genes. So a person can have 1 SMN2 genes or gene, or maybe 5 SMN2 genes, etc. The more the number of SMN2 genes, the more functional protein is produced. So you can imagine this as like in a normal scenario where an individual does not have spinal muscular atrophy, that the SMN1 gene produces proteins up to this level. And there's SMN2 gene again, which produces 10% of the total value of the protein, and it's somewhere right here. But the SMN1 gene is capable of taking care of the motor neurons and their survival. But when an individual is affected with spinal muscular atrophy, the SMN1 gene is mutated and this quantity completely goes down. So it only now depends on the quantity produced by the SMN2 genes. So it just basically varies that, if a person has 1 SMN2 gene, the protein production might somewhere be here. If they have 2 SMN2 genes, it might slightly increase. So it can go on increasing based on the number of genes the person have, the number of SMN2 genes the person have. Also, as this protein is important for the survival of motor neurons, the more protein produced, the more better the situation would be, and the number of SMN2 genes does affect the severity of the disease, and if the number of SMN2 genes are more, the disease is less severe in symptom, and its onset is also late. So based on the number of SMN2 genes an individual has and the developmental milestones they can achieve, spinal muscular atrophy is divided into different types. So let's look at them and also understand their characteristic symptoms. So there are 5 main types of SMA, the first being SMA type 0, SMA type 1, type 2, type 3, and type 4. It is important to note that SMA type 0 is the most severe of all and also exhibits the earliest onset of symptoms. And then as we progress from type 1, type 2, type 3, and type 4, the symptoms go on decreasing in severity and also their onset becomes late. Let's now look at all the different types individually. The first being SMA type 0. It is characterized by severe weakness, hypotonia, which is decrease in muscle mass, and respiratory distress at birth. There could also be a history of decreased in vitro movements, joint contractions, and atrial septal defects. The child might also exhibit facial paralysis and loss of response to stimuli. The child rarely survives past 6 months of age. The next type is SMA type 1. It is also called infantile onset SMA or Wernicke-Hoffmann disease. It is diagnosed before 6 months of age, in most of the cases around 2.5 months of age. The child here cannot sit independently. He or she would exhibit reduced muscle mass. Deep tendon reflexes are also absent. And the child might exhibit problems with sucking and swallowing. The child would usually survive only till about 24 months of age, only with supportive care. The next type is SMA type 2. It is also called intermediate SMA or Dubowitz disease. It manifests between ages 6 and 12 months. It is characterized by poor muscle tone since birth. The maximum milestone is to sit independently only with supportive care. But after 5 years of age, a slow decline in motor abilities is seen and the child would lose the ability to sit independently as well. Scoliosis could develop later in life and hand tremors are also present. Deep tendon reflexes are lost, and these individuals usually live up to about 25 years of age. The next type is SMA type 3. It is also called Kugelberg-Villander disease or juvenile SMA. It manifests after about 18 months of age. Here the individual can walk independently but might fall and have trouble with stairs. Fatigue is associated with day-to-day activities. Scoliosis might also develop as the disease progresses, and shortening of tendons is seen, which then doesn't allow joints to move freely. Gain in their motor functions until about 6 years is observed, but then after that age it starts to slowly decline until puberty. Here the individuals have normal life expectancy, it is not affected and is almost as equal to individuals who do not have SMA. The last type here is SMA type 4. It is characterized by muscle weakness, which usually develops only after the 3rd decade of life, that is after 30 years of age. Here the individuals are able to maintain their mobility throughout life. Life expectancy is again normal and accounts for about 5% of cases of SMA and is the rarest kind of these types. The common symptoms are muscle twitching, weakness, or breathing problems. Usually only the upper arms and legs are affected in this type. So there are also different types of SMA which are not associated with the SMN gene as we discussed earlier. So here for these types, the causative gene is not the SMN gene but is a different gene. So let's look at them briefly. The First one being spinal muscular atrophy, respiratory distress. It is also known as autosomal recessive distal spinal muscular atrophy. It is caused by a defect in the IGHMBP2 gene, which is located on the 14th chromosome. It causes a higher frequency of intrauterine growth, restriction, and premature birth. The second type is Kennedy's disease. It is caused by a mutation on the UBA1 gene, which lies on the X chromosome. The symptoms are mostly like SMA type 1. It usually appears between the ages 30 to 50 years. It causes muscle weakness as well, but the muscle weakness is mostly noticeable in the legs, arms, face, throat, speech, and swallowing difficulties, and major muscle cramps are observed. So let us now look at the inheritance of the SMA1 gene. It is autosomal recessive in nature. Before understanding this in detail, let's go through some quick basics of genetics. We have 23 pair of chromosomes, which means that in total we have 46 chromosomes, as you can see in this figure right here. So let us just focus on a random chromosome, let's say chromosome number 3. So here you can see that it exists in pair, and you should note that one chromosome of the pair is inherited from the father, and one chromosome of the pair is inherited from the mother in the child. Each pair has similar genes which could be different variants though. So what does this mean? It means that on chromosome number 3, if you can see on this particular position, or let's just focus on chromosome number 5 as of now, on which our SMN1 gene lies. So if the SMN1 gene is here, the SMN1 gene would be here again, but these two could be different variants. So the first one could be mutated and the second one could be normal, or both could be normal, or both could be mutated. So let us now look at the inheritance pattern of autosomal recessive condition, or SMA here in particular. Here we can see that a dad and a mom are carriers of the disease. A carrier is an individual who has one faulty gene and one working gene. So here you can see that there are 4 children, and as I told you before, that a child inherits one chromosome or one gene from the father and the other chromosome from the mother. So you can see that the first child here inherits the working gene from his father and a working gene from his mother. The second and the third child inherit one working gene from one of the parents and another non-functional gene from one of their parents. And the fourth child inherits, unfortunately, both the non-working gene, one from his father and one from his mother. So the first child here, who has both the working genes, he does not have the condition and is not a carrier as well. The second and the third children, who have one working gene inherited from their parents and one non-working gene, they do not have the condition but are carriers of the disease, which means that they can pass it on to the children, even though they do not exhibit the symptoms of the disease. And the fourth child, who has both the non-working genes inherited from his parents, has the condition. So there is a typical ratio that 75% of the children in this case do not have the condition, and 25% of the children would have the condition. And the reason why in 75% of the cases the condition is not seen or observed is because the condition would not appear in an individual if the individual does not have two faulty genes. And this is why it is called as autosomal recessive, because it requires two faulty genes to be developed in the body. Looking at SMA in particular, here you can see in the image that in the first child, who has both parents who are SMA carriers, The first child inherits one good copy of SMN1 from each of the parent. So he has two working genes and thereby he is unaffected. In the second and third child, they inherit one good copy from one parent and the other copy therefore is faulty, and thus they would be carriers of the disease. They would likely pass it on to the children, but they would not develop it. And in the fourth child, you can see that both the alleles of the gene inherited by the baby from both of his parents are the faulty ones, and thereby he would develop SMA. Another type of inheritance pattern seen for other diseases is autosomal dominant. Here, only one gene can cause a disease, but luckily it is not so for SMA. Let us now look at a diagnosis of SMA via different methods. The first method being the doctor questioning about the developmental milestones. As you might have observed in the previous slides, that a lot of our discussion revolved around developmental milestones achieved by the individual. So the questions a doctor would probably ask you is about if the children have problem with swallowing or moving around, sitting up, etc. And this helps them make a diagnosis towards SMA. The second being consulting a genetic counselor. It is very important to consult a genetic counselor because they help tremendously during the pre and post genetic testing phase, and also they could go through the family history and this way help you make a diagnosis for SMA by analyzing all the data. The third and the most important being genetic testing. As it is a genetic disease, it holds a very important value in making a diagnosis towards SMA. Here basically they take the DNA out from your body and they would check for the mutation in SMN1 or SMN2 or other pathogenic variants of the gene. And if they get a positive result, it would confirm the diagnosis towards SMA. The fourth and the fifth being MRI or magnetic resonance imaging and CT scan, which is computed tomography scan. These help to understand the muscle physiology and thus help make a diagnosis more clear. The last being electromyogram. So here doctor basically puts patches onto the child's skin and sends electric signal to the muscles, and this way they check if the signal is being conveyed to the muscles or not, and this again helps make a diagnosis more clear. Let us now at the end discuss the treatment available for SMA. The first treatment option is via gene therapy called Spinraza. It is used for both adults as well as children. It is given via 4 dosages in the first 2 months, and after that it is given once in every 4 months. Here what happens is that the protein production via the SMN1 gene, which is mutated, is increased. As more quantity of protein is produced, it thereby helps reduce the symptoms of the disease. The second treatment option is via a gene replacement therapy called Zolgensma. Here the SMN1 gene is replaced. It is usually done for children below the age of 2. It needs to be done just once in lifetime. And via research it has been proven that children who underwent this therapy have been able to reach or develop their developmental milestones better and in a faster way. Like they could sit properly, they could control their heads, etc. The next treatment option is via breathing support. So usually if an individual has problem in breathing, different machines or a tube would be inserted inside the nose which would help them breathe. The next treatment option is a diet plan. It is usual for kids to be underfed or overfed with this condition, so it's important to maintain a diet plan. And also if the child has or the individual has problem in swallowing, a tube inserted in the stomach could help. deliver them proper nutrition. The next option is via a brace to prevent the development of scoliosis, or a surgery in the later stages of life to treat scoliosis. The next option is via physical and occupational therapy. These could also include water therapy, or using different methods basically, which helps the muscles become stronger in day-to-day life. And we also have special tools like wheelchairs, for example, which can help the child or the individual to train their muscles and basically move around in a better way. At last, research on gene therapy and stem cells is currently going on so that we can find better treatment options for spinal muscular atrophy. That's it for today's video, guys. Thank you so much for watching this. If you found this video informative, please do share it. And if you have any questions, please feel free to comment in the comment section below. You can also find me on Instagram at ATGC of Genetics, and you could also mail me for further queries. Thank you so much.